So now we are going to switch gears, and we are going to start with the first session of lighting talks. For those of you who are not familiar, these are very short talks, 5 minutes, with 2 to 3 minutes question. And our first speaker is Jorge Cruz. Jorge? Jorge, where are you? Are you there? Okay, there he is. Now I am not panicking. He's a PhD student here at the Institute of Ecology, UNAM. And he works with fears, and he will tell us lots on their phylogenetic history as soon as we manage to solve a little issue. Don't panic.
Thank you. Para las preguntas yo voy a contestar fuerte. Para que no corra. Well, good morning. This is my presentation, Dissimilar evolutionary processes drive morphological and genetic differentiation with these collaborators. The isolation by distance is determined by increase in genetic differentiation among populations with increase in geographical distance as a result of reduced gene flow. While isolation by adaptation, gene flow among ecologically divergent habitats is reduced because of reduced starving success of immigrants from different environments. For example, we can expect this pattern: a positive relationship between geographical distance and neutral genetic differentiation, but in isolation by adaptation, we can expect no relationship between geographical distance and morphological traits under divergent selection, but a positive relationship between ecological distance and morphological traits under divergent selection. If we have two species, one of them with a restricted geographical distribution and a small population size, we expect that the neutral differentiation and phenotypic divergence are similar. But on the other hand, in species with a wider geographical distribution and large population size, we expect that the phenotypic divergence is higher than neutral differentiation. We studied two species, Abies religiosa, with a wider geographical distribution in a faja volcánica transmexicana in Mexico. On the other hand, Abies flinki, with a more restricted geographical distribution at the west of faja volcánica transmexicana in Mexico. We studied the leaves or needles in this case. We measure 15 variables, and then we select 3 for estimate phenotypic divergence, or PST. They are needle thickness. This is a cross-section of needle, abaxial distance, and number of raw stomata. On the other hand, we estimate GST of nuclear microsatellites. Results. We found not isolation by distance in Abies flinki, but we found isolation by distance in Abies religiosa. The relationship between morphological traits and climatic variables was dissimilar because while number of raw stomata was positive with longitude and isothermality in Abies flinki, but not in Abies religiosa, while needle thickness and abaxial distance was positive with precipitation of driest quarter, but not in Abies flinki. Then we contrast GST, or neutral differentiation, versus phenotypic divergence. These are the results. Always the phenotypic divergence was higher than neutral differentiation. But these are similar. Then the neutral processes are present for these characters in Abies flinki for row of stomata, needle thickness, and abaxial distance. While in Abies religiosa, we found a moderate, we found a signal of divergent selection. Conclusions. The phenotypic divergence is explained by neutral genetic drift and environment in Abies flinki, the same case for Abies religiosa, in needle thickness in abaxial distance. While number of row stomata, we found a moderate evidence for divergent selection. That's all.
Well done, Jorge. Thank you very much. Questions, please? Yeah, no?
In your first slide, you had isolation by distance and also isolation by adaptation. So I didn't understand, how would you differ between those two? How would you differ between the scenario of isolation by distance and isolation by adaptation?
We'll get another microphone at the lunch break, at the coffee break.
I expect in isolation by adaptation, the phenotypic divergence is higher than neutral differentiation for these traits. It was determined because the value of CH2. If the value of CH2 is closer to 0, we have a signal of divergent selection, but the value of CH2 is farther to 0, we talk about neutral processes. And the gray lines are confidence intervals. The red lines, confidence intervals for neutral differentiation.
And do you have a hypothesis as to why the number of rows would be adaptive and the other traits are not?
Yeah, the rough stomata are related to evapotranspiration. These are related with the precipitation in this case. But there are other mechanisms, for example, open or closed over stomatas.
We have time for another very quick question. Good. So thank you, Jorge, very much.